Good morning to everyone. It's only 9 o'clock this morning at this time. So let me thank the organizers, first of all, for really the kind invitation to be here and to actually the opportunity to present some of the ongoing work, current work of my lab, to actually exploit, of course, CRISPR-Cas9 tools for actually modeling first and then actually developing some possible treatments for neurological disorders. Well, the breathtaking sequence of discoveries on novel nucleases as well as novel methods to actually gene repair or base editing or gene expression modulation actually really provide an amazing arsenal of unprecedented tools to actually really repair a gene and its related disorders. But I think all of you agree with me that the brain is really a tough target to be actually therapeutically treated. And this for many different reasons. The first is the inherent complexities of this tissue, the fact that it's lacking regenerative potential, and that actually is strongly isolated by barriers from the rest of the body. Most also importantly, in neurons, the HDR mechanism is not functional. And even considering all of that, you have also to actually consider that if you repair the gene and you restore the function in neurons, if during the progression of the disease, the neuronal circuits which are embedded in those, in the neurons that you are treating, if this circuit has been deranged or lost during the progression of the disease, no matter that you actually restore the gene function, probably you will not get any therapeutic success in actually treating these disorders. However, I think the two seminal breakthroughs in the field had actually been achieved in treating and getting clinical success in treating neurological disorders. And the first is actually the lentiviral hematopoietic stem cell gene therapy for treating metachromatic leukodystrophies, and the second is the infusion of AAV particles in newborn with MSA. And these actually, in both these actually approaches, really provide a strong pool of evidence of arrest or strongly delay the progression of the symptoms in these two terrible, devastating infantile disorders. So I think these are actually of inspiration for all the field. I think that we can actually work on similar approaches with appropriate modification for also applying to other related neurological disorders. So today I'd like to tell you about two short unpublished stories from my lab. The first is actually related to Friedreich ataxia. Friedreich ataxia, or FRDA, is actually the most common inherited ataxia, which with an onset in early childhood and is leading to actually gait and limb ataxia, loss of postural equilibrium, which progressively lead the patients to actually being wheelchair bound. This disorder is also associated with severe cardiomyopathy and also diabetes, which leads this patient to have a reduced lifespan expectancy. The disease is actually due, at least the neurological disorder, by the specific loss of neuronal population. One is actually in the dorsal root ganglia sensory neurons, and the second are the Purkinje cells in the cerebellum. And my talk will be related to this, actually. And the causes of the disease basically is a nucleotide expansion. It's part of the disorder that Beverly in the first talk actually nicely presented to us, and in this case is a GAA triplet expansion in intron 1 of the gene frataxin. This expansion is actually in the patient is about 100 to 1800 repeats, while in normal individuals the repeats are lower than 30. And this long expansion leads to the silencing of the frataxin genes. How these silences is actually achieved by the expansion is not still yet settled, but actually one of the mechanisms that has been actually suggested is that this long triplet expansion in intron 1 is actually leading to a formation of a triple helical DNA structures, which is actually blocking RNA processivity. And this is actually leading then to actually a strong deposition of chromatin repressive mark like H3K9 or K27 trimethylation. leading to really a chromatin repressive state, which is associated with gene silencing. Well, frataxin is actually a mitochondrial protein residing in the matrix. It is essential for handling iron metabolism, and is also critical for actually the generation of iron sulfur clusters, which are prosthetic groups which have an essential function in chromatin mitochondrial complexes, and therefore frataxin mutant cells have an impairment in mitochondrial respiration. But also frataxin has also an important ferroxidase activity, generating ferric ions, and this is important to actually restrain the generation of radical oxygen species through the phantom reaction. So what we did in the lab is trying to actually model FRDA disease in vitro by iPSC technology. So we actually got fibroblasts from 2 different patients, one with a mild expansion, the second with a longer expansion, up to 900 GAA repeats, and we actually use non-integrating technologies to actually reprogram fibroblasts into iPSC clones. And then after having obtained these iPSCs, we actually decided to use the CRISPR-Cas system to actually delete the expansion in intron 1. We did this in 2 different ways. The first, we designed sgRNAs at the junction of the expansion in order to actually remove only the expansion by maintaining all the rest of the intron. With the second approach instead, we actually designed the sgRNA really at the 500 base pair upstream to the splice junctions between axon 1 and axon 2, and with this strategy we remove both the expansion and all the entire intron 1, which is a very long, is up to 10 kb. So we package we selected this sgRNA for the efficacy and for the low off-target predictions, and we clone them in vectors. We transfect iPSCs, and we actually screen for clones which have actually the removal and the excision of these parts in a homozygous state. So we did this, we select then the clones with the homozygous state deletion. And we make sure that we could actually having, after this genetic modification, iPSCs, which actually maintain morphology and critical markers of pluripotency, as well as correct actually karyotype. So then we selected 1 single clone for each of the deletion for each of the patient, making actually having at least 6 different clones as our collection of cell lines to be actually studied. And we also make sure that these clones do not have any type of mutation in the in silico predicted off-target sequences. So in Friedreich ataxia, in FRDA, the neurons which initially start to die and are lost in the early state of the progression are proprioceptor sensory neurons in the dorsal ganglia. These neurons are essential to actually contact the muscle, and they are essential to sense the muscle strength and contractions, and providing really a sense of the body position in the space. When actually these neurons are actually deleted, this leads to severe gait difficulties and instability that is typical of Friedreich ataxia. So we wanted to actually differentiate iPSCs in DRG sensory neurons. And to do that, actually, we recapitulate the milestone which happened during embryonic development to actually differentiate DRG sensory neurons, which came from the delamination from the dorsalmost part of the developing neural tube, where actually is present a strong wind signaling. So we actually devised a 3D culture system to actually differentiate iPSCs aggregates in dorsal ganglia organoids. We did so by actually exposing the iPSCs aggregates to a sequence of different neuralizing molecules, together with a strong inhibition of the GSK3 beta enzyme, and therefore a strong activation of endogenous wind signaling. This provided a really system to actually neuralize the iPSCs aggregates, and at day 20, the iPSC aggregates start to actually extend out of the aggregate aggregate long axonal tracts. And all these axons actually are peripherin positive, indicating that they have acquired a peripheral neural system identity. At day 50, most of these axons have really tremendously elongated in the culture, and we have signs of formation of synaptic contacts with the formation of punta positive for synapsin and V-GLUT1. So then we compare the 3D culture system that we have established respect to other protocols already published in 2D cultures, and to our first analysis we could actually say that the 3D culture system allowed really to actually increase and enhance the neuronal maturation of all the 3 different neuronal subpopulations, both proprioceptor, nociceptor, and mechanoreceptor neurons. And in agreement with this, more than 60% of all the neurons in the DRG organoids are actually able to fire repetitive action potential after current injection. But actually we were interested not only to have neurons, but to establish the entire connection which makes this neuron to actually contact the muscle. And in the muscle, these neurons contact the intrafusal muscle fibers, and when this axon contacts the muscle fibers, what happens is that, let's see if, sorry, oh my God. Okay, so when actually the proprioceptor axons contact the muscle fibers, they actually ensheath the muscle fibers, forming what is called the annulospiral axonal terminals. These are specific axonal terminals which identify the axonal connection with typical of the proprioceptor neurons. So we wanted to establish this connection in vitro. And what we actually did initially, we took a human myoblast primary culture, we differentiated them, and we have actually clear sign of differentiation of intrafusal muscle fiber, which is positive for these two markers, S46 and A4.74. And then we actually co-culture this primary myoblast co-culture with the DRG organoids. And what we saw is that 7 days after the starting the co-culture, axons from of the sensory neurons contacted the intrafusal fibers and actually made these spiral axonal terminals. And these are really specific for proprioceptor and not any other sensory neurons. So we thought that we have established in vitro a full connection between sensory neurons and their actually peripheral tissue. And in a triple immunofluorescence, we could also actually highlight the formation of synaptic contacts, carotene positive, between the axonal terminals and the muscle fibers. So then what we did, we used these protocols to actually generate the DRG organoids from the mutant and the isogenic deleted patient iPSCs. And then we took these DRG organoids and we profiled the level of frataxin protein. And as expected in the patient, DRG organoids, we have a strong reduction in frataxin protein levels that actually were rescued in neurons with either the small deletion, but even more with the long deletion. So we actually did not expect these results. Why, if you actually delete the entire intron 1, including the expansion, did you get an increasing expression of the rescue of the frataxin expression? So we explained this at the time in our minds by the fact that removing the entire intron, we also remove most of the repressive chromatin, which is actually embedded in this intron. And actually this is very difficult to remove in post-mitotic cells like neurons. So we thought that maybe this is actually the reason why taking out the entire intron will actually restore a physiological level of frataxin. And to answer to these questions, we actually profile the H3K27 trimethylation deposition in this area in the different class of cells. So as expected, basically we had that in patient FRDA neurons, we have a strong increase in K27 trimethylation in the promoter, axon 1 and intron 1. Compared to actually wild-type cells. In neurons with a short deletion, we have a reduction in these repressive marks, which were, however, was not complete. Only in the neurons with a large deletion, we have a strong reduction in the deposition of this mark in the promoter region and in the axon 1. So indicating that it was necessary to actually cut out the entire intron 1 in order to actually reduce the deposition of this repressive mark in the nearby region and therefore reconstitute the expression of frataxin gene. So then we ask if the reconstitution of frataxin expression in the patient mutant cells was sufficient to actually rescue any type of malformation that we can describe in the frataxin neurons. And indeed, as also published previously by other papers modeling this disease with iPSCs, we also found that axonal elongation from these mutant neurons is strongly impaired. So all the axons which actually extend from basically the DRG organoids were strongly impaired in frataxin mutant neurons. But when actually we, in neurons with the short or the long deletion, this extension was actually rescued, and in particular in neurons with the long deletion. Then we asked whether this also is also occurring when actually these neurons contact the muscle fibers. And in contrast, we have a nice formation of spiral axonal terminals, as you can see, which enwrap the muscle fibers. While mutant frataxin neurons, they actually contact the fiber, but does not seem to actually form the spiral connections. So they are impaired in really forming the entire and complete spiral connectivity. And this actually was rescued, at least with the neurons with the long deletion. Then we look for some markers, like for instance, mitochondrial DNA content is known to actually be reduced in these frataxin mutant cells, and this was actually rescued by the long deletion, in the long deletion neurons, and also expression of the aconitase gene, which is an enzyme which has prosthetic alpha-sulfur cofactor. Actually, the expression of these genes was actually restored, as well as also the expression of SOD1 and SOD2, which actually is increased because oxidative stress levels are probably increased in these neurons. Actually, the expression of these cells of these two genes was actually normalized in the neurons with the long deletion. But actually, what we cared was also try to organize these circuits in vitro in a spatially organized system. So what we actually optimize is actually to plating human neurons with their postsynaptic counterparts into microfluidic system, which allow really to have an ordered connectivity that we can then easily be studied. So what we did in this case, we have a microfluidic chambers with two lateral chambers. On one side, we actually plated the DRG organoids. On the other side, we plated the primary myoblast. And then these two chambers are connected up by microchannels, and in each microchannel, only one single axon from the sensory neurons can grow up to actually reaching the myoblast fibers, and therefore creating really a perfect and spatially organized system where actually we can study and we can image axons from the sensory neurons. So we did imaging of these sensory neurons of the sensory neuron axons in the microchannels, and we could actually find that mutant neurons have a reduced number of axons in the axons of the sensory neurons that actually was actually rescued upon deletion of the intron 1. So even the amount of content of mitochondria along the axon will restore upon a gene deletion of the frataxin gene. Right. So So next, we are excited about really trying to combining cultures of iPS-derived neurons in microfluidic system to organize neuronal circuits. Not only having a neuron, you need to have the counterpart in order to establish a specific connection to be studied. And we have also tried to actually expand this with actually generating and settling an in vitro system to actually generate the nigrostriatal connection between human iPS-derived dopaminergic and striatal neurons in the microfluidic devices. And I have no time to tell you about this story today. So conclusion of this first part is that we can actually differentiate iPSCs in 3D culture system generating DRG organoids, which we can co-culture with myoblast fibers and regenerate the full connection between sensory neurons and muscle fibers. And we can use this sophisticated in vitro system to actually probe for specific defects in human disorders. This is actually proof of principle with the Friedreich ataxia modeling in vitro with iPSCs, and we show that the excision of the entire intron is necessary for a full recovery of expression of frataxin, which allow the recovery of the neuronal defects that we have seen in this model in vitro. Let me go to the second story in the last few minutes, which instead is dealing with the DCAS activation system that actually Jeremy presented very nicely in his previous presentation. And we not did anything of so sophisticated. What we actually used was actually very traditional system of DCAS fused to an activatory system, a factor of transcriptional activation like the VP64 or the VP160. We use this as a classical system for DCAS-mediated gene activation. We wanted to try to actually explore some potential of gene modulation in the brain to actually treat a specific disorder. And we turn actually our interest to idiopathic partial or focal epilepsy because these actually are non-genetic disorders or diseases, and we wanted to see whether we could use a genetic tool to actually find a possibly therapeutic avenue for a non-genetic disorder. Indeed, focal epilepsy are the most common forms of epilepsy in adults. They are generally drug resistant, and the only way to treat this actually epilepsy is to actually resect the entire epileptic focus in the brain. But also the full resection of this epileptic focus might actually be not indicating in case this focus is actually very close to actually brain regions which are important for speech or memory skills. Therefore, in those cases, you cannot do anything with these patients. So we thought to actually tackle this important group of disorders. The idea was try to modulate the expression of actually a gene coding for an ion channel. In this case, the gene is called KCNA1, which is encoding for the voltage-gated potassium channel KV1.1. The expression of this channel is actually leading to an hyperpolarization of the neuronal membrane and therefore to an increased current threshold to initiate the action potential. Therefore, more you express this gene, more these neurons will be refractant to start the action potential firing. Once the firing is started, this neuron will actually have a normal repetitive firing. What is actually changing here is that you provide really a less likelihood of this neuron to start and initiate the firing of action potential upon stimulation of the expression of this gene. So what we did, as also Jeremy has nicely shown to you, is actually we designed sgRNA close to the proximal promoter of the KCN1A gene, and we found and selected one guide SG19 that was actually able to upregulate the expression of this gene by 3 to 5 times, both at RNA and protein level. We did this screening in mouse primary neurons because we thought that it's important to actually do the screening in a system that is close by to your in vivo application. This is much more hard, as actually Jeremy before actually alluded, using primary mouse cultures to actually test CRISPR-Cas tool. But actually, eventually, we did this, and actually, finally, actually, we got good results for that. Then we actually look for any type of targeting of this system by doing RNA-seq analysis in the transduced neurons, and we were delighted to see that in this RNA-seq global gene expression analysis, the only genes that was significantly upregulated was indeed the KCN1A gene. And no any other genes in the entire genome was actually found any difference. So with this data give us the confidence, so we move on in vivo. So the first thing we did is to use non-epileptic mice. In this case, we actually packaged the DCAS activatory system into two different AAV9 viruses, which are correlated together in the expression, and they get activated only by doxycycline administration. And we transduced animals again, kinase through 3 mice to express this DCAS activation system only in excitatory neurons in the hippocampus. And then transduced brains were actually taken out, brains sliced, and page clamp record. This was done in red fluorescent neurons that were those which were transduced. And we were happy to also confirm on brain slices that neurons transduced with this system actually have a reduction in firing, both in mean and maximal frequency of firing, and an increased current threshold to start action potential. Finally, we actually team up with Gabriele Lignani in Dimitri Kuhlmann's lab at the UCL in London, because they developed a nice model of temporal lobe epilepsy, where by injecting kainic acid in the amygdala, this leads actually to the degeneration of neurons in the CA3 hippocampal area. And this became the focus of epileptic seizure recurrent over time. So this is really a very good model of temporal lobe epilepsy in humans. So what we did now is to actually inject kainic acid in amygdala. A week later, we inject the DCAS9 activation system, either activating KCNA1 or a control activation system with the LAC-V sgRNA. And then we actually implanted a transmitter in these transduced mice for chronic video EEG recording in order to actually quantify the number of seizures over time in this animal. And then we leave this animal for 2 weeks, and then the other 2 weeks we provide doxycycline, and we in the meantime record all the seizures, so we could have a single animal in which we could quantify the number of seizures before the activation of the system and after the activation of the system. So we have the single animal could actually give us the difference of number of seizures upon activation of the DCAS9 activation system. And doing this actually work, we did a bunch of animals and record in chronicle way all the seizures. You see in green here when actually we administrated the doxycycline in the animals. So basically only those animals which actually have the DCAS activation system for the KCN1 gene have a significant reduction in the number of seizures over the 2 weeks, while in the control activation system, those animals did not see any difference in the number of seizures over time. And however, when the seizure was started, there was no difference in the length and duration of the epileptic seizure, which actually is really giving us the confidence that this is depending by the activation of a voltage gate potassium channel. The reduction of, this is the last slide, the reduction of the seizure was also leading to actually an amelioration of the behavior of the mice that we tested with the object localization task, and providing the principle that reducing the seizure, we can actually spare basically the memory and learning skills of these animals. I like only to mention to you that we also treated wild-type animal, and we showed that this treatment did not actually lead to any difference in the behavior of the animal. So here the conclusion are that we have developed a DCAS9 system that can actually be used, a genetic system, for non-genetic neurological disorders, like focal epilepsy, which are orphan of really a cure. Let me tell you that we could actually manipulate the neuronal activity both in vitro and in vivo, but actually this system requires the expression of the Cas9 over time to maintain the stimulation of the gene. So this might actually lead over time to actually some hurdles regarding the immune response of having a Cas9 to be expressed for such a long time to actually maintain the expression of this gene. So I finish here, and I thank the people who did actually the work. Piero Mazzara actually was a PhD, was responsible for the story on the sensory neurons in the lab. Now he has finished, and he's going to start his postdoc in San Diego. And the work on the focal epilepsy and DCAS activation were actually led in the lab by Gaia Colasante and her team, together with really the equal collaboration of Gabriele Lignani and Dimitri Kuhlman in London. So I finish here, and I thank you for your kind attention.